All right, so for this video, I'm going to draw the electron transport chain. So I've already covered glycolysis, Krebs cycle, and other videos. Right, just got to finish up with that electron transport, and then we should be good to go. Right, before we get there, we do have to do some accounting. So I need to know how many high energy molecules I have at this point. So we need to go back to our glycolysis picture here. Right, technically I'm going to make two NADHs from two NAD plus in this case, and two hydrogen ions, I guess. So I have two NADH molecules here. Then I go to my Krebs cycle, and I'm going to have a lot of NADHs in here, because remember, this is two pyruvates that are going to go through and do all of this, right? So I'm going to go ahead and use a different color here so I can keep track of everything. So I technically have two pyruvates, so I'm going to have two NADHs that are made here, two NADH made here, two NADH made here, two NADH made here, plus the two that were in my glycolysis picture. So at the end of the day, when I'm talking NADH, I have 5 times 2. I have 10 NADH molecules that I can use at this point, right? Those are high energy molecules that contain electrons that I'm going to be able to use in my electron transport. So I need to know how many there are, right? So I've made a lot of high energy molecules. I also made, it looks like, 2 FADH2 molecules in here, right? Now FADH2, as I alluded to beforehand, actually has a couple extra electrons in it. So 2 FADH2 molecules is basically the equivalent of 4 NADHs. But also, when I looked back through everything, remember I made a net gain of 2 ATP, but I'm going to lose those moving pyruvates into the mitochondria. I am going to have a little bit of ATP when it's all said and done, because I did get my 2 back in Krebs cycle. So I do have 2 usable ATP molecules, but I gotta some way, somehow turn these molecules into even more ATP, right? Ultimately, I'm supposed to be able to make 34 ATP in electron transport, and I'm gonna get all that energy from these NADH and FADH2 molecules. So again, just kind of counting, making sure I know how many of those high energy molecules I've made. Otherwise, the electron transport is going to happen on the inner mitochondrial membrane. And this picture is going to look pretty familiar, right? I'm going to have a membrane, right? So I got some phospholipids in here. Go ahead and draw those in a picture. And there's going to be a chain of a transport chain, a bunch of proteins I'm going to draw going down, and continue my phospholipid bilayer in here so I can see that this is still a membrane. And again, I'm going to take all of those NADH molecules, as well as all of those FADH2 molecules, which the numbers are on the other slide. I don't need to do that again. And I'm going to take all of those electrons and I'm going to drop them off into this transport chain, right? I don't have anything green here because I don't need anything green. I'm not absorbing energy from sunlight. There's energy in these electrons that are being dropped off that energy ultimately did come from sunlight. It was trapped a long time ago in photosynthesis. But all I need to do is drop off these electrons from these high energy molecules so they turn back into their NAD plus and FAD plus 2 counterparts. And those electrons are going to move down this transport chain. And this is very, very similar to what we had before, right? As those electrons move down this transport chain, they're going to power pumps that push hydrogen ion across the membrane. Now, which way that hydrogen ion moves doesn't really matter for our purposes, but let's say that it's over here and it's going to be pushed, taken from here, and moved down through this membrane, right? So just like in photosynthesis, I'm trying to create a really big concentration of hydrogen ions. And I'm doing it, excuse me, for the exact same reason I did it in photosynthesis. Somewhere else on this membrane, there's a transport protein, an open channel that will allow those hydrogen ions to flow back through. And it's the exact same protein that was used in photosynthesis. This protein is still called ATP synthase. And it's exactly the same. It functions exactly the same way. I take this hydrogen ion that's in high concentration on one side of the membrane, I let it flow back through that membrane, and I use that energy to power the production of a bunch of ATP. Remember, at the end of the day, I'm supposed to make 34 ATP from ADP molecules. And all of that energy is going to be provided by, right, NADH and FADH2 just indirectly, right? The electrons from those two molecules are going to help power proton pumps that push hydrogen ion across the membrane. So I push the hydrogen ion from one side to the other. I let it flow right back through an ATP synthase enzyme. And that's going to help me make a bunch of ATP. But I told you before that none of this would be possible without oxygen. So I need to put oxygen in this picture, right? I'm not going to be able to do any of this without oxygen, because oxygen is supposed to be at the end of this transport chain to catch those electrons that are flowing through that chain, right? I told you guys specifically, and we were, talking about a computer cord and how it has a current when it's plugged into my computer. But if there are no electrons to go, if I take it out of the computer, even if it's plugged into the wall, there'd be no current, because I'm going to need somewhere for those electrons to go. That's what oxygen's job is. It accepts electrons at the end of this transport chain. It will grab those electrons, as well as, a couple of hydrogen ions, and ultimately turn into water. So again, when I said that this is an aerobic process, I have to have that oxygen for this to function. And you have to have that oxygen. It's aerobic because without oxygen, you can't catch those electrons. If you can't catch those electrons, there's essentially going to be a traffic jam. The electrons that are supposed to be flowing down that electron transport chain can't move because there's nowhere for them to go. So this is the only aerobic step of respiration, but it's really important, right? Without it, I can't take advantage of all this energy that's in NADH and FADH2 molecules. I've made a bunch of those molecules in between glycolysis and Krebs cycle. So I want to be able to take advantage of those things. Otherwise, I'm just stuck with 2 ATP, right? Which sometimes your cells have to settle for that 2 ATP. The 2 ATP that you technically had when you finished glycolysis. So you're not going to do any of the rest of that stuff. So again, our electron transport picture should actually be pretty simple because it looks almost exactly the same as it did in photosynthesis. You just don't have any chlorophyll. And there aren't 2 photosystems because you only really need the one transport chain to power those proton pumps. So it should be a pretty complete picture. Again, I told you that respiration was the opposite of photosynthesis. The very last product of respiration is the very first reactant when I talk about photosynthesis. If I go back to my photosynthesis pictures, right? The very first thing I need to do is split some water to get electrons. Those electrons end up in high energy molecules, right? That's the exact opposite of what just happened here in the last step. High energy molecules dropped off electrons. They ended up in water. So if I go backwards, water, electrons, high energy molecules, everything pretty much looks the same. Then I go to my cycle here, right? I could take high energy molecules, right? Reverse everything here, make a 6-carbon molecule that could be glucose, right? Everything is the same, it's just backwards. All right, so that should be good for this picture.